Welcome to an early version of the show today on this Wednesday. Today we were very fortunate, of course, to be joined by Kelly Victory, as we always are on Wednesday. Dr. Jessica Rose comes back. She's a Canadian researcher. She has a degree in math, master's in immunology. She holds a PhD in computational biology, doctoral degrees in molecular biology. She has also a degree in biochemistry, and she has been looking at the VAERS data and very concerned about adverse events for some time. She has some updated data today on contamination of the vaccines. We are also very pleased to be joined by Dr. Joseph Fraiman, ER doctor, physician from Louisiana, who's been very concerned about the approval process for medication and vaccines by the Federal Drug Administration, the FDA. He manages Louisiana's Urban Search and Rescue Disaster Task Force, and he has led a group of physicians about these major shortcomings in the FDA's recent publications, and he has some recordings of his interaction with the FDA when he presented this data to them. We're going to get into that and more right after this.
Our laws, as it pertains to substances, are draconian and bizarre. The psychopaths start this way. He was an alcoholic because of social media and pornography, PTSD, love addiction, fentanyl and heroin. Ridiculous. I'm a doctor, for sake. Where the hell do you think I learned that? I'm just saying, you go to treatment before you kill people. I am a clinician. I observe things about these chemicals. Let's just deal with what's real. We used to get these calls on Loveline all the time. Educate adolescents and to prevent and to treat. If you have trouble you can't stop and you want help stop it, I can help. I got a lot to say. I got a lot more to say.
I think everyone knows the next medical crisis could be just around the corner, whether it comes in the form of another pandemic or something much more routine like a tick bite. You and your family need to be prepared. That's where the Wellness Company comes in. You know the Wellness Company. We have their physicians on, like Dr. McCullough, frequently. The Wellness Company and their doctors are medical professionals you can trust. And their new medical emergency kits are the gold standard when it comes to keeping you safe and healthy.
It's a safety net. It's an insurance policy
absolutely.
That you hope you're not going to
100%.
need, but if you need it, you sure as heck are going to wish you had it if you need it.
Be ready for anything. This medical emergency kit contains an assortment of life-saving medications, including ivermectin, Z-Pak. The medical emergency kit provides a guidebook to aid in the safe use of all these life-saving medications. From anthrax to tick bites to COVID-19, the Wellness Company's medical emergency kit is exactly what you need to have on hand to be prepared. Rest assured, knowing that you have emergency antibiotics, antivirals, and antiparasitics on hand to help you and your family stay safe from whatever life throws at you next. Go to drdrew.com/TWC. That is DRDREW.com/TWC to get 10% off today. Just click on that link. There are three reasons the central banks are dumping the U.S. dollar: inflation, deficit spending, and our insurmountable national debt. The fact is, there is one asset that has withstood famine, wars, political and economic upheaval dating back to biblical times: gold. And you can own it in a tax-sheltered retirement account with the help of Birch Gold. That's right, Birch Gold will help you convert an existing IRA or 401k, maybe from a previous employer, into an IRA in gold. And the best part, you don't pay a penny out of pocket. Just visit birchgold.com/Drew for your free info kit. They'll hold your hand through the entire process. Think about this. When currencies fail, gold is a safe haven. How much more time does the dollar have? Birch Gold has an A+ rating with Better Business Bureau and thousands of happy customers. I do not give financial advice, and previous performance is no guarantee of future performance. Visit birchgold.com/Drew to get your free info kit on gold. That is BIRCHGOLD.com/DREW. Welcome back. As I said, we got Jessica Rose and Joseph Fraiman in here. I want to bring Jessica in here first. As I said, she's a Canadian researcher, a degree in math, master's in immunology, PhD in computational biology, molecular biology, and biochemistry. And in addition to a little vaccine talk today, we're going to get an update about Maui. Please welcome Dr. Jessica Rose. There you are. Welcome back.
Hi. Thanks for having me back. Pleasure to be here.
You bet. Before we get into plasmids and all things vaccine, can you give us an update on what's going on with Maui?
Probably not better than anybody. I'm a little confused about what's going on there. It appears as though there's definitely something being covered up, according to what I'm hearing from locals that I know and and also don't know that you can hear on social media. They're talking, into their phones about what's going on. But what I keep hearing since the beginning is that there are children unaccounted for, and some people are claiming that they were actually stuck in their homes and perished the thought but burned, and other people are saying that they're missing. Like, I don't know what the truth behind that is, but nonetheless, on the subject matter, the most important things that I think we need to address are these events that led up to these potential deaths. Because if even one kid died, that's too many kids to have died, and people need to be held accountable. So whether it be leaving the fire before it was out to put out a fire on the other side of the island, to treasure the water, to not sound the siren because it wasn't an official tsunami, or because, they're saying that the fire started due to outdated power lines, etc. I mean, these are all things that, someone needs to be held accountable. That's
well, Jessica. I don't know on the government side. Unfortunately, they have these qualified immunities that allow them not to learn from their mistakes, while the rest of us take profound punishment when we make mistakes. But I assure you, I spoke to a lawyer over the weekend. The lawyers are not actually directly involved in taking action. They are not missing this opportunity, I assure you. I assure you there is a lot going on, and it will, and associated with those actions is a tremendous amount of private investigative activity, that sort of thing, sort of fact finding. So, and also you need to recognize that these attorneys that do this sort of work need every molecule of information, because they hold these people accountable to every single thing they find. And that's how they optimize their return on what they're working on. But it is on. So I don't worry, everybody. The legal system will have its way. The government, and their need for, I worry, when I saw Maui, I worried about our situation here in California, where we don't do forestry control. I used to look up in these San Gabriel Mountains when I was a kid, and they were crisscrossed with fire breaks. Well, we stopped doing that because they interfered with the immigration of a mouse. Some mouse wasn't able to get across properly, and so now, and they refuse to do the underbrush cleaning out, which they need to do. And when there is a fire, which there will be, it will be catastrophic here in Southern California. You would think Maui would be some sort of a cautionary tale. Nope. Because government, the individuals making the decisions, have qualified immunity, and they never learn because of that. They have no skin in the game other than re-election, which, again, in this state, we seem to re-elect incompetence. But I don't know, whatever. Who am I to say?
Yeah, the people who always seem to lose are the actual people. You know what I mean? It's like,
of course,
the people who are invested, the people who work hard, the people who are expected to go back to work even though they don't have a home, or they can't even go to see if there's anything left from their belongings. I mean, it's I hope there's some kind of justice here. I'm putting all of my thoughts and energies into that. So
I think there will be ultimately, but again, it takes time. But I want to get Joseph Fraiman and Kelly Victory in here as soon as possible. Before we do, what I'll do is I'll take a little break after we have this next topic, you and I. Give me what's going on. I keep hearing, not rumors, but I keep reading material about plasmid contamination by the vaccines. Explain to people what a plasmid is, what the risk of a plasmid is, and what the evidence for this is.
Okay, I'll try and do this very succinctly. Kevin McKernan is a genomics expert, and he found DNA in vials that he tested for some other thing. He was looking for a control. So this was not something he set out to do. DNA should not be in the vials. there should only be modified mRNA in the vials. That's the first thing. The way that the DNA got in is because the process that they're using to manufacture the modified mRNA for encapsulation in lipid nanoparticles uses E. coli, which have these circular plasmids in them that encode the spike gene. So it's a method for making a lot of DNA for in vitro transcription to make the mRNA. So it's something that we do, but we haven't done this in the context of modified mRNA with these 1-methyl pseudouridines. And so what we're thinking happened is at the last step, when they're supposed to purify the modified mRNA out using this enzyme that eats up the DNA such that you don't have any more DNA, was that the DNAs didn't work on what we're calling what might be hybrids. So the DNA might have fused to the mRNA, for lack of a better way of saying it, and that enzyme might not have worked to remove these things. So basically what happened is an hypothesis is that the DNA contamination in the vials that Kevin detected, and by the way, this has been reproduced in other labs, which is the way that we're supposed to do this. If somebody finds something, you have to reproduce the results. This is the most important thing in science. So this has been done. But this is the working hypothesis as to how this DNA actually got brought over. It got mixed up in the mRNA because it wasn't properly taken out during the purification step and encapsulated in the lipid nanoparticles. So that's all. Kevin and Philip Buchholz is one of the other confirming cancer specialists. He has his own lab as well. Separate vials, completely independent investigations found very, very similar things, which is this DNA contamination. So there are many repercussions of this, of which we might be able to explain in some of the adverse events we're seeing. So that was a long summary, but that's that's what's going on.
We got it. We got it. So there's a couple of questions. The one we have for the average person, what is it about the DNA that is associated with adverse events? That's sort of one topic. And then I was thinking about the biology of the DNA plasmids unwinding and opening and fusing with mRNA. Do they have a theory about how that happened?
Well, you have to linearize, like basically you take it out of its circular shape before you do the in vitro transcription. So there's this, I don't do this, so I've never done it in myself, but this is how it's done. So you put it into its linear form. So somewhere along the line, we are hypothesizing that that's what happened. So DNA RNA hybrids are a thing, and they create these little things called R loops, which, I never mind what that is, but these can also create problems in vivo in terms of creating genomic instability. So
genomic instability in human cells, other human cells.
Yeah. So interfering with normal processing in terms of what happens genomically. So the other thing that I've heard in one of the conversations I've had with colleagues is that these little bits of DNA, they're not circular plasmids that are being transferred. They're like little bits. And according to one of these experts, these little bits more easily can integrate into the genome of humans. And the problem with that is, multifold. They can actually get inserted inside very important genes. So say you get insertion of this little piece of DNA into an existing gene, you're going to mess up that gene's function. So
Sure.
again, this is all just our line of thinking right now as to the possible ways that this could go wrong.
But back to the R loops, you don't really even need that. If you're interfering with DNA repair, that's sufficient to cause massive issues.
Mm-hmm. Yeah. Everyone, if you want to know more about that, you can go to the Nepetalactone newsletter, that Kevin's newsletter, and he wrote an article called DNA RNA hybrids, R loops, and nuclease resistance of the mRNA vaccines. He explains this really well here, and there is a Nature paper that was published called R loop derived cytoplasmic RNA DNA hybrids can activate an immune response. So basically, these things they're a part of like nature. Like it's not a weird thing that R loops are created. The problem is that they can accumulate, and that can cause problems. Again, Kevin is the one to answer these questions in more detail. But all of these all of these issues arise because of. Sorry, go ahead.
I was just going to say, I know also you're concerned about lipid nanoparticle polysaccharide contamination and changes as well.
Yeah, so that was another issue that was of concern because of this process 2 method of, for lack of a better way of saying it, growing up the DNA using E. coli. So we do this, like it's not something that's like, oh, we're using E. coli. That's not the big deal. The big deal is that when you use this as part of this process, you need to make sure that you don't take lipopolysaccharide, which is, it's a part of this E. coli with you in your final product. So we always test, or, the people who test for these things test for endotoxin levels when they have a finished product to make sure that they're not exceeding some kind of accepted level. Because if you inject someone with lipopolysaccharide, you're probably going to induce anaphylactic shock or septic shock. It's pretty bad. So it's interesting because Kelly actually mentioned, that she has a PEG allergy, and if she got injected with these things, she probably would go into anaphylactic shock. So it got me wondering that. Because we have seen a lot of reports of anaphylaxis following injection with these things. So I'm wondering if it isn't partially the PEG and also maybe the lipopolysaccharide. I don't know. We need to confirm all of this, but these are the hypotheticals that could potentially go wrong. And this is why we measure these things, not we, me and my colleagues, but the people who test for contaminants before they inject things into people, for example. Like you have to do this as part of good manufacturing practices and responsibility and all that stuff. So yeah, be interesting to see what the redacted levels of the endotoxin are.
Right. Have they been doing that? And by endotoxin, let me just explain to people that these E. coli, it's how they create the diarrheas and they create bad illness and things. They release this lipopolysaccharide that's an endotoxin that's part of their makeup. But have this is this being done appropriately, that they're testing for these things?
Well, the only document that I've seen disclosed by the SpikeFax, or in the context of the SpikeFax product, where they did measure endotoxin levels, the measurement was redacted. So that really blew my mind, because I don't understand why a test result would be redacted. I can't even speculate. I mean, I've thought, well, maybe the reason why they're redacting it is because it was above some accepted criteria and they didn't want people to know, because it's not like they haven't done that before. But again, that's speculation. Yeah, it would be really interesting if we could prompt people who have labs to actually reproduce all of these findings that Kevin and Philip and everyone are finding, because this affects everybody. It really does.
Yeah. Yeah, this would be amazing if they could do that. But I'm just astonished at the way people feel they can hide information and not invite worst case speculation. I just don't understand people. Do people not understand how it induces paranoia and speculation when you hide anything? That's how human brains work. And if your thinking is a little more paranoid than not, you can go way all over the place and fill in the unknown. It's sunshine, fresh air, conversation, discourse, repetition of results. That's why the scientific method, right, exactly. That's why it's always been since Bacon. That's the way it's been done. And now all of a sudden, and by Bacon, I mean Francis Bacon, not pork. But that's... he's really the first example of the experimental method, the scientific method, and it's always been done that way until the present moment. This is what people have got to understand. All right, and to that point, we're going to bring Joseph Fraiman in after the break here. Jessica, you're going to stay with us. We're going to bring Kelly in as well, Dr. Kelly Victory, of course. It's Wednesday, and I think we'll probably kick off with a conversation about the shortcomings of studies and randomized controlled trial. We'll be right back. Fall is right around the corner, which means dry, flaky red skin from allergy season is coming with it. But the best way to take care of your skin is with our skincare secret, Genucel. You don't need to worry about that puffy, tired eye look or those annoying dark spots or even dry, flaky skin because Genucel Skincare has you covered. Susan and I love our Genucel products so much, we want you to try our personally curated skincare bundles. It's risk-free at Genucel.com/Drew. Genucel works so well, you can see the results in this unplanned live moment on our show when the Redness Repair Cream repaired my skin in just minutes right before your eyes. Their concentrated vitamin C serum helps keep your skin plump and hydrated. Plus, with their immediate effects, you can see astonishing results in under 12 hours. Quick, effective, and easy. Go to Genucel.com/Drew right now to try our bundles and save over 60% today. And remember to enroll in Genucel's world-class concierge program for additional savings and free shipping. Don't wait. It's Genucel.com/Drew. Genucel.com/DREW. I want to share with you a teeth whitening system that goes beyond merely enhancing your smile. Primal Life Organics Real White Teeth Whitening System offers convenience and rapid results without harsh chemicals. Light, blue light for whitening, red light for gum and oral hygiene, and you can just do both if you wish. Works naturally, promoting gum healing, tooth remineralization, gives you a brighter and a healthier smile. Again, no peroxide involved. Consistent usage yields remarkable results. Take this opportunity to transform your smile and at the same time, optimize your oral health. Aim for 5 times a week for the best outcomes. Discover more about this remarkable teeth whitening system and other products at drdrew.com/primal today. That again is drdrew.com/primal. Be sure to use that link for 60% off. drdrew.com/primal. Do it today for 60% off. Some platforms have banned the discussion of controversial topics. If this episode ends here, the rest of the show is available at drdrew.tv.
There's nothing in medicine that doesn't boil down to a risk-benefit calculation. It is the mandate, public health, to consider the impact of any particular mitigation scheme on the entire population. This is uncharted territory, Drew.
And here we are, Dr. Kelly Victory. I give you Jessica Rose, and after a few minutes, we'll bring in Dr. Fraiman.
Terrific. Hey, Jess, welcome back. So happy to have you
Hi,
here.
Kelly.
Let me just follow up on this issue about polyethylene glycol, PEG. I had huge concern about that from the very, very beginning as somebody who has a very severe, long-standing allergy to polyethylene glycol. It turns out that that's a very common allergy. PEG is used, a lot of people don't know this, in topical agents all the time. It's in a very commonly used, for example, in cosmetics, in sunscreens, lotions, that kind of stuff. And that for me, and probably I'm guessing for the majority of people with that allergy, doesn't cause concern. It's only when you take it orally And I found out the hard way, taking a medication that actually went under your tongue, and the carrier was polyethylene glycol. And I developed a Stevens-Johnson's type reaction and was very, profoundly ill from it. Yes, profoundly ill. And it turns out that that's not that uncommon. I can't imagine what would happen to me if I injected it. That's when I put it under my tongue. If somebody injected it into my arm, I can only imagine. And that's why I found it so stunning that physician after physician and nurse after nurse was perfectly happy to have me injected with it when I was in the hospital, telling me that, don't worry, you're here. We can take care of you if you become deathly ill. Anyway, kind of these are different times. I want to change gears for 1 minute before we bring Dr. Fraiman on. Jess, you posted a link to an article or a story about this study done in the UK looking at all-cause mortality in people vaccinated versus unvaccinated people in the UK. Is that something that you feel comfortable talking about? I won't do the study justice, but it looked at rates, all-cause mortality rates, and broke it down based on vaccination status in the UK. And it's a large group because they were looking at people over the age of 18, which in the UK is about 50 million people. And it appeared to me, based on what you linked, that study showed pretty clearly that the vaccine itself was the cause of increased mortality, nothing else. Is that a study you can talk about?
It's probably a cross post. So I'm not really the all-cause mortality person, but yeah. So, but I would comment that the weirdest part about it is the absolute lack of possibility that the increase in deaths, which doesn't have any known cause, could absolutely not be due to these products that were rolled out in 2021. So yeah, that's the part about it that really boggles my mind. I mean, something is killing young people, healthy people. There are more people dying across the board, and this is data. I mean, this isn't. It's nothing more than data that's being collected. So data doesn't lie. People lie. So something has to explain this, and the timing is unusual, and makes it suspicious. So it's that's what I would say about that. I mean, actually, somebody asked me the other day. It was a really good question. It was a spin-off of an interview I saw with Catherine Austin Fitz. And she made a really interesting comment about how a lot of the adverse events that we're seeing can kind of easily be written off as kind of the end game of somebody's existing condition. And I've been calling that symptom laundering. And I think that she's absolutely right. For somebody who's older or who has some kind of condition, an autoimmune condition or cancer, it seems like these things, for all intents and purposes, are absolutely ramping up all of these conditions. It's got some kind of immunological dysfunctional node. But of course, not even the people are making the association back to the shots. They're just thinking, oh, well, this is just the disease course running its course, and this would have happened anyway. So I thought that was really interesting. So somebody asked me about that, and they said, well, what can we actually do to jostle people, to engage them into starting to think that maybe there's a connection between this and this? And, well, go outside of that realm of possibility where this is just something, a problem that you've already had that is, following through. Look at kids, look at healthy athletes. I mean, we already are, but it's very, very important that we push this, because there's absolutely no reason for a child, for example, who's healthy to succumb to any kind of severe adverse event after getting an injection, unless it's because of the injection. So there's no way to argue with that. So yeah, it's
to wit, I would say, the concept of this new diagnosis, sudden adult death syndrome, SADS, as if this was something that existed prior to the COVID vaccinations. We have had, they made up this new category called sudden adult death syndrome. What are you talking about? That honestly, that diagnosis did not exist.
What are you talking about?
You're saying, what are you talking about? And people started throwing it around as if, SADS. I'm saying, no, I don't. It's a made up diagnosis that didn't exist prior to the need to have something to label all of these people who are suddenly dropping dead with no other explanation. So I know that we want to bring Dr. Fraiman into this conversation as well, but I will tee it up in this way before all four of us start talking. There's no question that we have seen this unprecedented number of adverse events. There's been a lot of daylight thrown on, concerns about the trials, concerns about pharmaceutical companies, owning the scientific journals. Lots of interest in or conflicts of interest and those sorts of things being exposed. But suffice to say, this didn't just start in the past 2 or 3 years with COVID vaccinations. We have all become, certainly I and Drew have become much more aware of it, much more cognizant of the absolute ownership of the scientific journals by big pharma, those sorts of things, the way that the FDA and the CDC are corrupted, the amount of money that they're making on patents that they own, and on and on and on. But suffice to say, this is not something that started yesterday. And so I want to kind of have that as the backdrop for the conversation that we have going forward for when Dr. Fraiman exposes his conversation with the FDA and more the information that you, Jess, are throwing on these studies and what we're learning about these particular injections, that this is not something that is new. And I think for me, it's the consummate existential crisis as a physician, because I really have to wonder to what extent have the therapeutics that I have used, the scientific studies that I have relied upon, the constructs that I have, that were foundational, which of them really are on faulty, really shaky ground? Because we have exposed an awful lot in the last 3 years that isn't particularly flattering on the industry of medicine.
Well, I would say that. Really, go ahead, Jessica.
Go ahead, Jess.
I just wanted to throw in, it's absolutely true, and it's kind of a mixed blessing that this has happened to shed light on these problems that are inherent in this entire system, this industry, which is supposed to be about health. But this mRNA stuff, the modified mRNA and LNP platform thing, it really needs to stop, because the trend now is that because these things have allegedly been proven to be safe and effective, and I'm being sarcastic here, they have not been. They've actually been, we've provided very strong evidence as a scientific community that they're not either of those things. And further, follow-up products, which are going to be used in humans for cancer therapies. You watch for this, it's coming. For other types of viruses, packaging, all-in-one products using this mRNA tech, animal use. I mean, it's all coming. And the problem is going to be there aren't going to be any trials. There aren't going to be any tests done anymore, because it's proven to be safe. If people are just listening, I'm doing air quotes there. It's absolutely not the case. So this is the danger. We really need to ding, ding, ding the bell on these particular products and on the inherent problems. Yeah.
No, you are spot on here, Jess. And I think that's a point that needs to be hammered home. I frequently get criticized when I say that these aren't safe and effective. People say, mRNA technology has been around for 15, 20 years. I'm saying, you're right, it has. Let's be real clear. It's been around.
Been studied.
And it has failed, and sometimes miserably, with tragic results where all of the test subjects died. It's not that we haven't tried to create a safe and effective mRNA shot in the past. It's that they have failed. Okay. They have failed over and over and over again. It has not been proven. And you are absolutely right. They have done, sleight of hand. They have led people to believe that these vaccines are safe and effective. And therefore, the platform is now safe and effective to use going forward. And they're going to use it for everything. And I've said from the beginning, this was their goal all along. This was always their goal, to make mRNA a household word, to make people believe that it was a platform that was absolutely tried and true, nothing to worry about, and that people would accept this genetic modification as something that we've always had, in the way that you accept other vaccines or other technologies. So I think that to me, Jessica, is the single biggest risk of all of this, independent of all of the horrific adverse events we've seen, is their ability to pull the wool over the eyes of people to believe that this platform itself has been safe and doesn't need further testing.
Well, let's get to the RCTs and the flaws in the studies and whatnot. We're going to bring Dr. Joseph Fraiman in here.
Actually,
but I want to say what?
Sorry.
Kelly,
wait, he's back now. Sorry, he had stepped away. I was looking at his camera and he stepped away, but he's there now. You can continue. Sorry.
Okay, good. But I was going to say, Vinay Prasad has been complaining about these inadequacies in what comes to market for quite some time. And I'll say that, if there are issues with the mRNA technology, so be it. If somebody has a terminal illness and there's a possibility that delivery system could help somebody with a life-threatening condition and turn that around, is worth the risk. Is it worth the risk for a healthy 19-year-old to use that kind of? That's a wholly different question. Unless we understand these things clearly, we're not doing our job. So speaking of that, Dr. Fraiman has been worried about this long before we got to the table, Kelly, and he has been talking to the FDA about some of the shortcomings. Dr. Fraiman, welcome back. I'm going to turn it over to Dr. Victory, but let me, before I do, just say, could you just sketch for people again a review of your review of the data and what you found?
You mean from our study that we performed on serious
from your study,
events?
Yes. What was different in your study versus what was reported by the FDA?
A quick summary of it is we reanalyzed the original trials from the messenger RNA vaccines, where Pfizer, Moderna. We identified that in the Pfizer trial, initially there was an increase in serious adverse events, and it has to do with how you count them. There was more events in the vaccine group than the placebo group. If anyone counts them, you will find that it's really not a debatable fact. The next part of our study was we looked at a list that was created by a group called the Brighton Collaboration, which is a World Health Organization endorsed list of something called adverse events of special interest. And what this is are adverse events that are prior to the clinical trial. They said, look out for these. These are potential harms to look for. So we then took that list and said, are these increased in the messenger RNA vaccine clinical trials, to get rid of statistical noise, so that things that were probably not related to the vaccine would go away, and we could focus in kind of like a magnifying glass on these adverse events. And there we find that when you combine Pfizer and Moderna, they both appear to have an increase in these serious adverse events of special interest. And together it's quite clear the rate is about 1 in 800, one serious adverse event for every 800 participant in the original clinical trial, which is, I believe, about a 40% in that range, a 40% increase in these serious adverse events. And then the last part of our trial was we looked at this increase in serious adverse events versus in the clinical trial, how many people were prevented from being hospitalized by the COVID-19 vaccines, by the messenger RNA vaccines. And we found that for both in the Pfizer and the Moderna trial, it appeared that the messenger RNA vaccine was causing more serious adverse events than it was preventing. And I just want to be clear, there's lots of limitations on how to interpret that last part of the harm benefit analysis, because, for example, if the trial had went longer, it's possible there would have been more hospitalization reductions. And if the trial was done in older people, maybe they would have found more hospitalization reductions. So it would have shifted it into another direction. And then on the other hand, if you did the trial in younger, healthier people who had been infected before, or if the trial maybe happened during a different variant, such as the one currently being experienced, where the hospitalization and death rate is much lower for each infection and the vaccine's less effective, there the harm benefit would actually be worse. And so you would actually end up with even the ratio would end up being thrown in the other direction. And that harm benefit analysis is it's fragile, is I guess what you would say, in that things can throw it in either direction, I think is the best way to interpret it. And that's the summary, a very quick summary.
And what were the special interest adverse events? If you could just sketch out some of those for us. I'm wondering if it includes some of the things I'm seeing. It's what's interesting, because I'm seeing neuropathies and POT syndrome and arrhythmias. And is that the stuff that was a serious special interest?
Yes, all of those would have been included, I believe, onto the adverse event special interest list. From our trial, what it looks like is that it would be causing a very rare harms in many different regions, many different body systems. And so that when they add up, because if you have 10 different serious adverse events in 10 different causes, 10 different ones at a rate of 1 in 10,000, those add up, and then you have a serious adverse harm rate of 1 in 1,000. So lots of serious adverse events, they add up together. And what we saw in our trial, that it's not more than 50%, it's probably 30-40% but it was the most common are in the coagulation disorders, which are blood clotting and bleeding. Those are the ones that were the most increased in the vaccine group compared to the placebo in Pfizer and in Moderna. But it's not that their rate's so crazy. It's 3 in 10,000, 4 in 10,000. And so, but then you add on these other ones, all the other random systems, like you just pointed out there, and together, that's how you would get that 1 in 800 number.
I'm trying to make sense of it. I've said many, many times that I think the adverse events, to me, we are having a pandemic of the itises. Itis meaning inflammation. It's pericarditis, neuritis, nephritis, hepatitis, colitis. All of the itises. It is an inflammatory process. I'm not sure how the coagulation issues fit into that, what it is about the inflammation. The clotting cascade was not my strong suit in medical school. So
I have a theory. I have a theory.
Yeah.
My theory from the beginning. My theory from the beginning has been that this is an endotheliitis, and the endotheliitis is interacting with the bloodborne elements. That's been confirmed by a couple of people that I've talked to. Yeah.
And so again, that would fall into the itis, the pandemic of the itises. It's an inflammatory process. In that case, an inflammation of the lining of the blood vessel that then attracts platelets and serves as a nidus for a blood clot to form. So I think, Joe, you can verify this, that the adverse events of special interest, I think, mostly fall into, again, those inflammatory processes. There's something about these spike proteins or the spike proteins in conjunction with the lipid nanoparticles that causes this gross inflammatory process.
Yeah, that was the general concern that started our, this project, was the discovery that the spike protein is causing endothelial dysregulation. And I mean the spike protein from the COVID vaccine, which is coincidentally the same spike protein, messenger RNA, that we put into the vaccine. Even that they're not the same, but if they weren't the same, then the vaccine wouldn't work. So it's a little confusing, the messaging on this, but the spike protein itself has been shown to cause endothelial dysregulation. But so yes, and the blood system, it pretty much connects to every organ system of our body. So I would imagine the organs that are at the highest risk of endothelial dysfunction would be the ones that would end up suffering from such a problem. But I don't know the mechanism. That's not what our study was, not mechanistic based. Jessica could probably give you mechanism much better than I could.
Well, I could comment. Marc Girardot, plugging him here, has done a lot of excellent work on this. And there's no doubt that this is about which cells are actually getting transfected, because whichever cells, and more likely, I'll just add that on, these are going to be the endothelial cells lining blood vessels. Mm-hmm. So you have entry of this product into your body, either intramuscularly, it's going to hit the capillaries, or maybe you're going to hit a vein or an artery. And in any case, it's going to enter the blood system. It just matters what the concentration, or as Marc would put it, whether or not a bolus forms. Bolus is just like a ball. So if you have a concentrated amount of these lipid nanoparticles, and we're talking about like trillions of them apparently, in a very small vessel like a capillary, the likelihood of transfection is going to be really high just because of the conserved space. So if you have a punch of transfected cells, like in one region, then you're going to have all the things that come with that. You're going to have your spike protein translated, churned up, mounted on MHCs, CTL. Sorry, I'm saying things that nobody knows what the words mean, but you're going to have an immune response mounted against those cells that put flags on their surfaces saying, hey, I have a foreign protein on me, and they're going to get destroyed. So if you have a big local destruction, that's going to cause your clotting pathway to get disturbed, overactivated, and clots are going to start to be thrown. And that's how you're potentially going to get necrosis later on down the line. So that's what Marc has actually written a lot of articles on this, and I prompt everyone to go read them. He makes a lot of sense on a lot of this, and it really is just it's like you said, it's about the circulatory system reaching every single part of the body, all the tissue, and it's just a numbers game. Like where is there a concentration, and is there a concentration of these lipid nanoparticles getting to somewhere? Could be the heart, could be the brain, it could be the wherever.
Right.
And of course, they're going to get there in the circulatory system. So, yeah. Sorry, I'm taking all the talk time.
No, yeah. So I want to take this topic and say, okay, I'm just sitting here watching the screen myself and saying, here we are, four scientists who all have done the deep dive on this over the past 3.5, almost coming up on 4 years now. We all have a pretty deep understanding of the mechanisms of the rate of these incidences, all the risks. We know all this. Okay, let's change gears. Turns out we have some agencies in the United States whose job it is to know this stuff. Let's start with the FDA and the CDC and the NIH. So what I want to talk about, and I'll throw this first to you, Joe, if we know this, what did the FDA know? When did they know it? Okay, and what did they do about it? Because it's implausible to think that just the four of us are so darn smart that we're the four who figured it out, and those poor, people slogging away at the FDA, just none of this crossed their radar, and they have no reason to know this. Let's talk about, from your perspective, Joe, what did the FDA know? When did they know it? And what did they do about it?
I actually, I'm all for, giving the FDA problems when they're doing a bad job. And I think that the reason we bring this up here is essentially was a discussion that my group of authors from our study, we presented our study prior to publication because we kept getting rejected. So we thought it was important for the FDA to know about our study. And they called us in for a meeting with essentially all of their top officials of biologics, basically for vaccines, vaccine safety. And we ended up having a meeting with them. And I think that, there was a large amount of the, even these top officials who I do think were actively concerned and looking for if the harms were true. And the issue that I felt in much of our discussion was that they need confirmation that the harms are absolutely 100% true, and before they're going to pull the lever. And so in statistics, right, you have we say things are statistically significant when they're 95%, when we're saying it's 95% certain it's not random. But for harm, in my opinion, if we're 80% certain that a thing's going to harm a population, or 90%, or I think that we maybe need a different way of judging these things for harm versus benefit, because we don't want to give out a drug that's not beneficial. That's pretty clear. We want to be real certain, because if you give out a drug that's not beneficial, you're only going to deliver harm. So you want to wait for that. And but I believe that the FDA has kind of resulted in their way of evaluating harm based off of our meeting was that they presume that the null hypothesis is true, that there is no harm until they can absolutely demonstrate it, and they can demonstrate it over and over again. And the problem
I have two questions. Yeah. Hey, go ahead and finish your thought, but then I want to ask a question. Go ahead. The problem?
The problem is that the harms that we're most concerned about, so you have myocarditis, right, which we know the vaccine causes, but it's a very, very rare event. I've seen it once or twice in 10 years as an ER doctor. And so, if I may, it's so that's not going to, you're not going to really see that so much increase. But imagine if you increase the heart attack by 10 or 20%, right? If you increase a heart attack by 10 or 20%, that's going to cause 100,000 heart attacks in the country. But
right.
It's really difficult to see that. Imagine even just this is a way of explaining kind of why it's so hard epidemiologically, but as an ER doctor, I'll maybe see one heart attack a month. So let's say 12 a year. If we increase that 10%, 20%, we're talking about I'm going to see still about one a month. And I wouldn't notice that difference in terms of my day-to-day activities. And it turns out that when you try to do this in big epidemiological studies with millions and millions of people, you're not ever going to see that. You will see it. And then on the next study, you won't see it. And then you'll see it again. And then you won't see it. And our surveillance system's not designed to pick up something like that. It's not possible for it to pick up small increases in pretty common causes of serious harm.
But since you had direct interaction with the FDA, which I have not, but it seems to me that you've laid out a fundamental flaw. If their approach fundamentally is we won't act until we have proof that harm is being caused, can you imagine using that same thing if you're a manufacturing facility making a baby stroller? Or if you were an auto manufacturer making a car, if you were building a bridge, and you had to prove that that the wheels actually fall off the
stroller. I would argue that this is a wholly new position by the FDA. It seemed to me that they were overly cautious. No, it's not.
No, I do not believe this is a new position by the FDA. This is. I mean, even from the approval standpoint, the original concern that I was pushing was this efficacy, that you don't want people to take a drug unless we have certainty of efficacy. And we would imagine that how the FDA standard is supposed to be, you're supposed to have 2 randomized controlled trials, double blinded, and they're supposed to find an outcome that's clinically meaningful. But if you go and look through the last 10, 20 years of FDA approvals, you will find a ton that are not blinded, a ton that are not randomized. There's not even another group.
Placebo controlled.
not
Right.
even placebo controlled,
Right.
and their
No.
outcome is not clinically meaningful and not replicated, and these drugs are still been getting approved. So this is the COVID vaccine trials actually, in terms of if you look at the last 20 years of drug approvals, is actually not, it's definitely not the worst. It's far from the worst.
Right.
We've actually had many more drugs. And then the other part, what I was talking about there, is our surveillance system, which is supposed to be looking out for harms. And the problem is that if we don't do good, really well-controlled clinical trials before we approve these drugs, we end up in this situation where we live in a world of uncertainty, and we have to rely on a lot of observational data where sometimes we can figure out some serious harms and we can identify those, and then we withdraw those drugs. But once a drug is approved, we can't do randomized controlled trials on it anymore. And many of those drugs just live in uncertainty. And that is our current situation. And the COVID vaccine is an example of this, in that we don't know if, because they didn't study hospitalization reduction specifically, they didn't study it over 2 to 3 years. I take it like this, right? I remember, Drew, you said last time that you believed that overall it was beneficial in the elderly, but you were uncertain in the younger, healthy population. That's a fair belief.
Correct.
It's a belief. You don't know. You don't know that because there wasn't a trial that we have to rely on. And imagine like a hypothetical experiment was done, and the COVID vaccine, placebo-controlled, double-blind, randomized controlled trial went on for 2 to 3 years, and it ended today.
Mm-hmm.
Now, imagine that. So everyone in this vaccine trial has been infected with COVID, multiple times, some of them. Now, who gets hospitalized more from everything? Because I imagine in this trial, the vaccinated group would get boosted whenever the CDC decided randomly that it was the time for them to get boosted, which they have done, which they do. They just make up a time, boost now, this group boost now. And if we just listen to those, so we did it perfectly. Would the group that got the vaccine end up being hospitalized less when you take into account all the boosters, the reduction in COVID hospitalization that looks like it probably happened in the first 6 months, but less clear after boosters? So did it just delay hospitalization for 6 months? The answer is, actually, do any of you guys have an answer? Or do you think you know how that trial would fall out? Like
It's a brilliant thought experiment.
Well,
Go ahead, Jess.
the
What
reason
would
I
you
get
say?
bogged down. Go ahead, Jess.
I just think that's a brilliant thought experiment. I didn't even extend my brain into that arena. But yeah, I imagine that, from what I'm seeing in the peer-reviewed literature and on the ground and from case studies, the people who are multiply injected would certainly represent most of the hospitalized people. But what an interesting thing that would have been. And also, if they hadn't injected all the placebo participants as well, that would be helpful.
and it's a reasonable opinion. It's your belief. And the point of what I'm trying to say here is that, and we could have someone else here who says, I believe that it would have had a great reduction in hospitalization down to the 1-year-old. And that's their belief. The reality is we are uncertain to the answer to that question because we didn't answer it. And we're never going to fully 100% know. Yet the problem here that I think exists probably within our population is that there's a group of people who are confident. Like you said, I don't know. I think maybe we're going to have more hospitalizations. And I'm sure, Dr. Drew, I would presume, say, I think we'd see a benefit in the elderly. I'd guess Dr. Victory will say, I'm not so sure we'd see any benefit. I think it might be overall harm.
Right.
Correct.
But you would have a voice of uncertainty in what you're saying. The problem here is that there's a huge percentage of our population who's confident and certain that everyone ages 6 months and up would have a hospitalization reduction. This group of people, which includes our government, are irrationally confident. They're an irrationally confident group. And now there's also, on the other end of this, there are groups who are confident that if this study was done, that every single person within this trial, every single age category will be harmed. This group is also irrationally confident. This is the majority of our population of this country is irrationally confident. And so I apologize because I've definitely just offended a large portion of your audience on whichever one they are. But it's a
but you're right.
Statement.
But this is why this is why the average vaccine takes and should take 6 to 8 years to come to market, because people shouldn't have to guess. They shouldn't have to come to their own, what's your best guess conclusion? We should have this data. We should know what does happen to pregnant women when you inject them with this. We should know, what do you actually decrease transmission by this thing? We should know, what is the impact of multiply boosting people in rapid succession? Those are questions that are normally figured out during the 6, 8, 10, 12 years worth of testing that's supposed to happen before you launch something on the public, and certainly before you start doing something as unprecedented as mandating it for people against their will.
Mm-hmm.
Yes.
Joseph, can I ask you what you think would have happened if we'd let this actually run, and also what your opinion on why it wasn't run for 3 years, for example? Do you believe it was an emergency story?
If it was run for the entire time, what I believe is that it likely would have increased hospitalizations in below people 65, but and in the older group, I initially thought that it would probably have reduced hospitalizations. However, now when I think about it over a 2 to 3 year time period, I think it may have possibly delayed it for 6 months, but I'm not so certain of the hospitalization and death reductions that our observational studies are suggesting, because in the clinical trial, we didn't see for once people were infected within the vaccinated group, in the clinical trial, we didn't see that those people got hospitalized less. That doesn't appear to be the case. And they also didn't appear to die less. So when it stopped preventing infections and that became clear, and then it started just not preventing infections, but preventing hospitalization, I can't say that that's not possible. I just don't see any evidence from the clinical trial. So I question if that actually occurred, or if it was basically a classical observational bias that happens, called a healthy user bias or healthy vaccinee bias that tends to be seen in vaccine studies, where you end up seeing a mortality benefit or a hospitalization benefit in the group that got the vaccine. And so I just don't think that we could rely on. I don't think that we could rely on observational data for efficacy of drugs. That has classically been failed. That's been a classic failure of it. Every time we do this, we end up making a massive mistake. And it just happened time and time again through the history of medicine, of observational data, misleading us into believing something's working when it isn't.
Let me throw a wild card in here, Joe, as well. And would it affect your guess as to decrease in hospitalization rates, even in the older people, if we had allowed people to be treated? If we'd actually treated people with the readily available, safe, and effective medications, everything from, steroids to ivermectin to all the things we could have done, rather than
antibiotics.
Antibiotics, so that people weren't dying of bacterial pneumonia that could have easily been treated because it was simply called COVID and they were put on a ventilator and allowed to die. These are, would the fact that, because I would submit to you, and again, we're all guessing here because the study wasn't done, But I think any positive benefit of the vaccines would be eliminated had people actually been allowed to be treated with the medications that we knew from the very beginning would have been very, very effective.
It's a different study, though.
I would push back, and I fall into a category of people who've questioned the vaccine, and I hold these alternative treatments to the same standard that I hold the vaccine, in that I need to have the large scale randomized trials to really fully evaluate it. But I'm sympathetic to the use of incredibly safe medications in people who are for conditions that are potentially deadly. I do that in emergency medicine, and but without full evidence-based trials, of course. But do I say for sure that I know which medicine's working and which one's not? But I'm sympathetic to using very safe medications that can be potentially life-saving. Right. But I'm not willing to say I'm certain that any of these alternative medications that are plausible, had we studied them better, that they offered a benefit. There's hints
Joe,
that they may be good, but
could you go? I want to make sure we get this in before we run out of time, which is this uncanny procedure the FDA has in terms of determining certainty of adverse event in relation to a vaccine. And I'm wondering, you have some recordings legally recorded of your interaction with the FDA. I wonder if you actually have the recording where they describe this guy. They got a guy that goes and determines whether something is or is not. It determines the impossible, whether something is or is not related to the vaccine with certainty.
Here, I'll play the first thing here, which is what they said regarding. Here, I'll just play this here.
You've analyzed the total number of events rather than the number of people who had at least one event. So if you look at those tables at the top, it's sort of number of people with an SAE.
Now, this is the first thing that they brought up, the first critique of our study. They said we analyzed the number of events versus the number of participants. This is seems like a statistical argument that's of irrelevance, but turns out that the FDA, for all of its previous drug studies, they don't count the number of serious adverse events that happen to people in a clinical trial. They only count the number of people who have them. So if you're a person who has multiple things, and especially imagine in a trial where you're given 2 doses of vaccine 1, vaccine dose 1, dose 2. If you have if you have a heart attack after dose 1, then you have a stroke or another heart attack after dose 2, that's counted as 1. And now the fact that the FDA just doesn't count the number of events at all, there's no reason to not also count that. I'm not saying that counting the number of participants is bad. No. They're both useful and give different information. FDA doesn't do it. And I would put out for anyone who's listening, if you want to go back through FDA trials, because they have not done that in any of our prior trials. And that is a much more sensitive. It will catch more events. It will catch more harm because people who are sensitive to a drug's serious adverse events, turns out they're probably sensitive to more than one. That's just how medicine works. If you're going to get one bad one, you're probably more likely to get another bad one. And that has been not what our FDA has been doing, which concerned our group, I would say, our entire group. And they also, I will continue on here from the FDA's meeting here, and they didn't take here. I will play this video, this recording here.
We don't do statistical significance testing of raw numbers of AEs because we don't consider that informative for safety for multiplicity reasons, and also because, for instance, if you look at the list of AESIs, there are many things that are related and could be different manifestations of the same phenomenon. And then some of these things are completely different. SOCs are unlikely to arise from the same mechanism of action, and lumping them all together for a significance test isn't necessarily.
So the purpose here of what they're saying here is they're not doing statistical testing, not statistical testing on on adverse events. They are seriously. This is really insane to me.
No kidding.
He seriously is saying there, we only just look. If there's a huge difference that's so obvious that we don't need a statistical test, then we say, okay, that's a big difference. Let's look into this. That's absurd, because then he goes on to say that the way that they look at these adverse events is they look at each one, and then there's a guy. This guy, that was the one who's saying this, that when they identify them, they try to connect it. They go, okay, is this in the right amount of time from the vaccine? Do they have any comorbidities? Maybe that's what explained this. But when you're using a novel vaccine, how you're supposed to know what serious adverse events are coming? And this blew the minds of all of the researchers I was working with, physicians and scientists, that the FDA was doing this in such a sense. Here, this is one of my co-authors questioning them on this, about not using the fact that it's a randomized controlled trial. You've randomized these two groups. If there's more of something in one of the groups, there's one reason why there's more. It's the thing you use, the medical intervention. That's the whole reason we do randomized controlled trials. When you do observational studies, when you find the difference between groups, you're always like, oh, is it something else that we don't know about, that we're not thinking about? It's called a confounder. It's the reason observational studies are so unreliable. Here, they're evaluating randomized controlled trial data the way you evaluate observational data, which is insane. Because I think I said this the last time I was here. A Vioxx heart attack. Someone has a Vioxx heart attack. They come into my ER and they were taking Vioxx, and how do I know that heart attack was from Vioxx, or was that just a regular heart attack? And they would have had a heart attack if they weren't taking Vioxx. The only reason we know it is because in the randomized trials, more people who took Vioxx had heart attacks. That's all we know. It wasn't like the placebo had 0 heart attacks. And only, so right. This is one of my co-authors here trying to hit this point home here.
An event very likely to have occurred to them in any case, then that event is less likely to be related to the investigational product.
Right. It sounds like what's done in pharmacovigilance. But how do you then leverage the fact that you're dealing with a randomized trial?
I'm not sure what you mean by that. Again, using hypothesis testing to look for a causal relationship between the investigational product and placebo, again, is typically not done for a variety of reasons, one of which is the multiplicity concern, because
So he's saying there, multiplicity is the reason they don't do statistical testing. We are not doing multiplicity, saying if you test 200 different serious adverse events, you're going to find a false positive. He's right. We just combine them all. We ran one test. It's one test, all of them combined. And then if it's increased, you know you have a problem with your drug. You don't know exactly which one of these adverse events is causing it, but you know there's a problem if the vaccine group has a higher rate of them. It was very, very concerning. This entire meeting terrified my entire work group. To be fully honest, they went on to explain to us that they then began to say that their rationale for not being concerned was essentially because of real world data didn't confirm with what didn't connect. And if you believe in evidence-based medicine, that's not really how that works. If you find it in the clinical trial and the observational data isn't connecting it, that's a problem with your observational data, not the clinical trial. So they need to re-look at this. And here, this is.
I guess a question for me is that, and I actually bring this up for our folks just so that maybe Steve and Rich can comment more on this, is that your thesis that seems to me, if I understand it correctly, is that more people experience serious adverse events. There was a greater number of serious adverse events with the vaccines than there were people who were prevented from having serious COVID-19. And that seems to be a little bit at odds at what my understanding is of understanding the global real world evidence and post-marketing surveillance. So
that's a real big disconnect from anyone who understands evidence-based medicine, that you're going to compete your they use the term real world data. I don't know when this term came into popularity, but real world data is called observational data. When I went to medical school, and I was taught it's not that reliable. Yet real world data sounds great. It sounds, it's the real world. That's the real thing. It just turns out it's not that reliable. And then we have this problem that I was
not only that. Not only that, but we spoke to the Danish researcher that published that observational real-world data on lots having more adverse events than other lots. Other certain of the manufacturing seem to be associated with much higher adverse event rates. Could not get that published for 2 years, right. And since published, completely ignored. So there's some weird bias within the real-world data that's concerning as well.
We were challenging them on that if their surveillance data is adequate. And one of my co-authors, who's a rheumatologist, a pediatric rheumatologist, had this to say. It's quite a taking.
This is just an N of one, but February 25th, I filed a VAERS report on a 7-year-old patient who had a cardiac arrest following his first Pfizer vaccination. It was about 30 hours after he got the vaccine. I didn't receive any follow-up about it. The patient died about 8 days later. I submitted another email offering to update the report with the death of the patient. It's been 2 weeks now. I still haven't heard anything. So I'm wondering if there's more extensive surveillance than just VAERS-based surveillance for cases like this. And I would think that if the death of a 7-year-old following the vaccine is not, meriting follow-up, either the system is totally overwhelmed or there's something wrong with the reporting system.
I want to play you some of the responses to this, because we're explaining to them that there's just from this man's, was one physician's who just happens to be within our study group. This is his own experience with the VAERS experience with VAERS, and here's what they have to say.
Such interest, we're happy to make sure that it didn't get waylaid somewhere. Usually, the pediatric deaths, I know that they have been very good about chasing down. I know because I've heard details of each of them in many cases.
That doesn't mean that you. This is the worst response you could think. This man saying you didn't follow up on a pediatric death, and I want to finish this
word
thing.
Salad.
The FDA, instead, what this should have been like is this is a holy shit moment. Leaders of the FDA, guy saying it's been 2 weeks since a kid died, and you haven't contacted me saying, okay, we are going to figure out what went wrong here. We're going to fix this problem. You should have been contacted. Instead, we're getting things about, email us. Just write a huge study and then get a meeting with the heads of the FDA, and then you can email us about something. But here, let's go on.
Does that sound right, Steve?
Yeah, so they should be followed up within a matter of days, actually. So just drop me an email or Rich, and then we'll just send it to our surveillance group.
That's our surveillance
to make sure that it didn't. I got it fall through the cracks.
It did fall through the cracks.
My guess is, if anything, it's just that they're behind. But I can't believe I can't believe something like this would fall through the cracks.
You can't believe something like this. It did fall through the cracks. You just heard about it.
I'm pointing fingers, but I think that the larger issue.
And just to put that,
very reasonable.
Yes.
I should say, just to put this in some perspective, just a couple weeks ago, the FDA reprimanded and shut down a baby formula manufacturing facility because 4 children were hospitalized and 2 of them subsequently died. Okay. That's tragic. Don't get me wrong. Tragic. But they shut down an entire manufacturing facility and pulled the product from the market because 4 children were hospitalized. Compare that to the tens of thousands of reports that we have on VAERS, which we know is grossly underreported, by the way. The V-Safe, they pulled that entire surveillance system off and stopped it. In fact, one of you should talk about that. The V-Safe system, which was kind of the backup specifically geared towards the COVID shots, where VAERS is, takes all comers for vaccines. The V-Safe was, again, not my system. I didn't create it. None of us created it. It was created by them specifically to look at adverse events related to these vaccines. They have abdicated their responsibility to follow these up. If this doesn't piss off every American, then you are asleep at the wheel, because what you are exposing, Dr. Fraiman, I think is horrifying. This is the state of what we've got for the FDA. God help us.
Yeah, no, I have much more just recording that is terrifying. It's a terrifying thing that
and
I
Joseph,
don't believe. Yeah, go on, please.
It's. I just want to tell you that this is common. I have regular conversations with the woman who created Open VAERS. We're always going back and forth on this data, and it's a thing that if you file a report successfully into the system, which is a big deal, and it doesn't always happen for everyone that they go from having a temporary ID to a permanent ID. But if you try to do a follow-up report, i.e., if you originally filed for myocarditis and then that person died, it will never get done. It's almost a thing that you can say about VAERS, and it's true what you said. It's like, this is either most inefficient system ever, or it's just kind of inherently designed that way. And I don't know why. I mean,
Yes, I can explain. I think that my concern personally is that we have a system here of the FDA. The FDA is responsible for approving drugs. Peter Marks, who was in that meeting with us, is a head of biologics for vaccine safety.
He was there?
He actually is. He was in that meeting. Yes, this was the top. Oh, this was a top level of a meeting of directors of biostats, directors of. This was from the directors across the biological departments, and what he has his signature on the approval of a booster. I don't remember which one, but he has his signature on it. Now, the same people who are approving drugs are responsible for checking if they made a good idea, if that was a good idea. That is a serious conflict of interest that we've had built into our system. The CDC is making policy recommendations, and then they are the government of studying if their policy recommendations were a good idea. We have built a system that is totally not making sense. And then to cap this off, imagine that we allow these pharmaceutical companies to do their own trials, that they collect all of the data instead of someone like the NIH being responsible. Board government workers just being unbiased, saying, hey, does this thing reduce hospitalization? I don't know. Let's see. Or it's the most perfect use of bureaucracy. And we can figure out if things work or not. But if you use a pharmaceutical company which pays a research, clinical lab to research this, which gets a new contract each time, as long as it keeps finding good results,
right?
You're going to end up with not necessarily honest reviews. Like when I buy a Samsung TV, I don't read Samsung's reviews of the TV. I
right.
look to see independent reviews.
You don't?
The way we are accepting drugs to be allowed into the population is essentially just reading the Samsung review that this has the best picture, the best sound, and that is just a really serious issue that is easily preventable. We have the personnel the researchers to do these trials properly and figure out what drugs work and what don't. We have all the resources. We know exactly how to do it. We're not doing it. We are allowing corporations who have criminal records for lying to us. We're trusting them.
Right.
Our system has major issues, and people do need to be scared, because this is the COVID vaccine. If this is the thing that brought people's attention to this issue,
Right.
Go and look through all of your drugs and see how they were approved, and go imagine doing the same amount of research onto every drug that a person's on, the same way that you've heard about the COVID vaccine. And I can assure you, it will not bring you confidence for many of them, especially anything that's of the last 20 years, approved in the last 20 years.
No, and I think that really, I'm cognizant of the time here, Drew. I think that this brings me full circle to the thing that I really wanted to focus on, which is that as bad as these COVID vaccines, in my estimation, are horrific. They should have been pulled from the market, I believe, immediately. They should never have gone to market. But this problem far, far supersedes, it goes beyond what happened during COVID. As you're pointing out, Dr. Fraiman, this is something that has been longstanding, decades in the making. It's one of the things that Bobby Kennedy Jr., regardless of how you agree with him on other things, climate or otherwise, he has absolutely made a commitment to get to the bottom of this regulatory capture, the fact that we have pharmaceutical companies in bed with our agencies whose job it is to protect the safety and security of the American people, I think is a problem. Our scientific journals have been captured. You can't rely on anything you read in the what I thought were the storied medical journals, and on and on. So I think we are exposing a way bigger problem here that's going to need to be addressed.
I remember Bobby's call is when he gets, as soon as he gets to office, where to get into office would be to call the majors publishers in and to threaten them with a RICO action if they don't get their act straight. Joe, you were going to say something?
As before, what I was talking about, our majority of our population being irrationally confident in their viewpoints. At the other hand, I believe there's hope in that because there's a minority of this population that is rationally uncertain, and they are the counterpart to the irrationally confident. All three of you are in that rationally uncertain group, and you all have probably slightly different opinions, and that's totally okay. That's normal. And when things are uncertain, we're going to come to slightly different places in what we think is probably true. But the problem is, it's uncomfortable for most people to be uncertain, but people have to choose. Do you want to be rationally uncertain, or do you want to be irrational? Because if you want to be confident that you know what's going on, you then have to also, you're choosing to be irrational. And we need more population in this rationally uncertain group so that we can possibly change this system to make it work for us, to protect us the way that it's supposed to. These agencies were built to protect us, and they should, and they can. We have the way, we have the means, we know how to do it. And we can do it.
I want to add something in. I want to add something very important in here that was taken away by COVID, and that is the individual's practitioner's clinical judgment. Joe, I don't know about you, but I see lots of COVID, and it gives me a certain clinical sense of what's going on. By the way, I'm seeing the new variant come on board, and it's quite different than the previous infections, and it has remarkably responsive to Paxlovid, strangely. But I don't use it because it's so mild. It only lasts a day, maybe 3 days, and can be kind of intense. And when it is, I use the Paxlovid. But it's a totally different syndrome I'm seeing. That is informing me. That's helping me make decisions about vaccines or further vaccine therapy or treatment. The individual practitioner's clinical judgment is a feature of our system that is being systematically taken away by physician extenders, by centralization of decision making, and this is a major piece of our problem that doesn't even come into this conversation or the data you've brought in. I just wonder if you have a comment about that.
Yeah, I also I personally haven't hospitalized a patient with classical COVID-19 syndrome since February 2022. So it's been about a year and a half, maybe a bit longer. Is that Yeah, since Omicron essentially came into effect, I haven't hospitalized a single patient. So I've hospitalized lots of patients with COVID. You know, they had schizophrenia, maybe with a psychosis episode, or they were a guy who missed dialysis, and it was like, "Oh, do you have a cough?" Yeah, I do. Now that you say it, but this syndrome changed dramatically from what it was before, and it's and we don't have a way
it's changing again.
It's
It's changing again, and. Was that natural immunity? Was that natural immunity plus vaccine? Was it vaccine? Was it vaccine alone? We don't have any way of parsing these things out, or less pathogenic organism, period. You know, it could have been just less pathogenicity, period. But we can't parse these things out because they're not doing the studies.
Yes, we know for certain, yes, but the thing changed in the solid change from when it went to Omicron.
Right.
It can't be that everyone just was infected. There's something. It stopped being a problem. The disease changed dramatically to go to zero hospitalizations. Anecdotally.
Remarkably, it did exactly what every other virus has done from the beginning of time as it mutated. It became more transmissible, more contagious, and at the less time, less lethal, less severe. It would be reportable. It would be reportable for a virus to mutate in the other direction. I'm not saying it's never happened in the history of the universe, but it would be reportable.
It would be weird, but based on those FOIA documents we saw yesterday, maybe this isn't something from nature. just saying.
I will tell you. Interestingly,
know what it would do.
Interestingly, and I have said this from the beginning, because this was not from nature. Let's be clear. It was not. This was a lab-created virus. But fortunately, 100%, you heard it here first. 100%. But I will tell you,
I agree.
The good news is, in all seriousness, is that despite the fact that this was a lab-created virus, it mutated in ways that were exactly based on what a naturally occurring virus would do, which was great, because had this started to mutate differently, if they'd found out some way to embed components in it, to splice pieces in that made it mutate differently than that, meaning those two things, becoming more contagious, but at the less time, less severe, we would have been in a heap of trouble as a species. Yeah. Fortunately, that's not what happened, despite the fact that it was lab created.
Everyone's been very, very generous with their time. We are way, way over. But Joe, I'm going to ask for one more thing. We really haven't highlighted the guy, the FDA, how they decide if something is a Vioxx-related event or not, a vaccine-related event or not. Do you have that on tape where he goes, "Hey, we got a guy named John." John goes out and decides. John the plumber. John the plumber goes out and says, "Is it a vaccine reaction?"
Let me see if we got this here for you. That him stating that exactly in those sense, he gets to it over and over, but using randomization.
That was the most to me of today's been a lot of astonishment, but that was the most astonishing thing from our last conversation. I want to put that out, finish with that. That's our sort of return to tonic with a flurry.
This dude Bob, who did
I got it here, I think.
Very likely to have occurred to them in any case, then that event is less likely to be related to the investigational product.
So we played this earlier here.
I'm not sure what you mean by that. Again, using the randomization to do hypothesis testing to look for a causal relationship between the investigational product and placebo, again, is typically not done for a variety of reasons, one of which is the multiplicity concern, because we may have a case where there's no difference in total numbers of AEs, but a substantial difference in a specific SOC, which is causal. And you can't go around testing every PT, every SOC, and have believable hypothesis test results from that.
I appreciate that, but I think we're not looking at so much as hypothesis testing, but even just the notion that given you have a randomized trial, you could start with very crude level, looking at all cause and looking at AESIs. My impression is that's what the AESI list was indeed created for, is to have a better sort of a focused look at and gene hypotheses about what we might see?
I think the AESI list, the Brighton list, I believe it was intended for primarily post-marketing surveillance.
No,
but I
not true.
Could be wrong about that.
You are.
The critical review memos.
So I'm sorry. This whole recording is a lot of boring talk between people who like statistics a lot, and
yeah.
But the content of it is explained with going into each individual case and looking at all these individual things. I get, next time I come on here, I will exactly identify these things, because it's so boring to replay that I feel bad.
That's a date. That's a date. You have a date with us. A date to return and play some highlights. But I thank you both for spending time with us today. You've been very generous and been very insightful. A lot of interesting things have come up. What we're supposed to do is raise questions, raise issues, and I think that's a lot of what, and then focus on weaknesses in the process. And that's what we're supposed to do. It's not like we're doing something nefarious by doing this. That is our job and has always been our job. Kelly, you want to wrap up too?
Yeah, I would say that we have got to return critical thinking to medicine and certainly to these regulatory agencies. It is their job, as I said, to protect the American people, not come up with every which way from Sunday to explain away things. It's as if this is sort of a Saturday Night Live skit. If it weren't so tragic, these guys, I've met guys at the Jiffy Lube who have better critical thinking skills than these folks in the FDA boardroom. So thank you for sharing that, Jess. It's always great to see you. Love your insights on all of this and your scientific detail here. So thanks to both of you for being here. Sorry for taking up so much of your time today, but I think this was a great conversation.
Agreed.
Pleasure.
We will see you back.
Mechanics know everything.
Yeah, they do. They do.
And people don't question them the way they question us. Okay, guys, we'll wrap it up with that.
More bad is bad is usually, that's pretty clear. It's easy to understand concepts.
Exactly.
All right.
We'll be back on Tuesday, I think, by Jessica. And Kelly, are you with us on Tuesday next week? We're out tomorrow.
I am not. I'm back. I'm back. No, I'm back. I'm solo on Tuesday with Brian Hooker, who wrote,
that's what it is. Yes, that's right.
Yeah, Brian Hooker, who co-authored a book with Bobby Kennedy Jr. About the vaccines. So I am doing a one-on-one with him on Tuesday the 12th. And then we've got my my pal John Phillips I think you're back on Wednesday with John Phillips.
Yep.
We're going to be talking on Wednesday. And then we've got Pierre Corey coming back the following week.
Great. We'll see you all with Kelly on Tuesday and me and Kelly on Wednesday. We'll see you on Tuesday. Thank you for joining us.
Thanks.
Ask Dr. Drew is produced by Caleb Nation and Susan Pinsky. As a reminder, the discussions here are not a substitute for medical care, diagnosis, or treatment. This show is intended for educational and informational purposes only. I am a licensed physician, but I am not a replacement for your personal doctor, and I am not practicing medicine here. Always remember that our understanding of medicine and science is constantly evolving. Though my opinion is based on the information that is available to me today, some of the contents of this show could be outdated in the future. Be sure to check with trusted resources in case any of the information has been updated since this was published. If you or someone you know is in immediate danger, don't call me, call 911. If you're feeling hopeless or suicidal, call the National Suicide Prevention Lifeline at 800-273-8255. You can find more of my recommended organizations and helpful resources at drdrew.com/help.
I lived in Dallas, I lived in San Francisco, I lived in Miami, New Orleans.
You look old enough to have lived in all these cities.
Man, I am 56 years old. No, I'm not. No.
I was like, tell everybody that.
It's crazy because
I tell everybody that. It's very impressive.
I feel like you almost believe me for a second.
I did. I was like, wow, shit.
Like, black people can say anything about age. White people is like, man, I got to take this. I believe it.
Absolutely. I was like, man, that is good genes.